This is a poster presentation titled Novel Protein-Protein Interaction Tool Defines Mismatch Repair Deficient Breast Cancer as a Distinct Molecular Subtype. Mismatch repair, or MMR, alterations are important prognostic and predictive biomarkers in a variety of cancer subtypes, including colorectal and endometrial. However, in breast cancer, the distinction and clinical significance of MMR is largely unknown. This may be due in part to the fact that genetic alterations in MMR genes are rare and only seen to occur in around 3% of breast cancers. In breast cancer, most clinical emphasis is placed on a patient's individual hormone receptor HR status, ER, PR, and HER2, as well as Ki67. Most recently, genome-wide association studies have grown in popularity as it offers an approach to investigate complex diseases such as breast cancer. However, GWAS has fallen short in identifying missing heritability. Our fundamental understanding of the complex architecture of the genome due to additive genetic effects is not fully represented by GWAS. Analyzing the complex architecture could demonstrate subgroups of patients which share variants in genes in specific networks with shared phenotype. To combat this problem, we recently developed Proteinarium, a multi-sample protein-protein interaction tool with the ability to identify clusters of cancer patients with shared gene networks. In the present study, we analyzed the Cancer Genome Atlas TCGA data using a multi-sample protein-protein interactions analysis tool, Proteinarium, and showed the distinct separation in the MMR deficient and intact specific networks in a cohort of 996 breast cancer patients, including 89 MMR deficient patients. Proteinarium, a novel multi-sample tool used to identify clusters of patients with shared PPI networks, was used to compare the MMR deficient and MMR intact patients. MMR deficiency was defined as having a mutation in one or more of the 8 MMR machinery genes. The seed genes top 100 upregulated genes for each patient were selected using Z-score data from the RNA-seq data. Proteinarium uses the experimentally validated PPI information from the string database to build network graphs based on the seed genes for each patient. The similarity between each pair of network graphs is calculated using the Jacquard distance, which is recorded in a similarity matrix used as an input for clustering graphs. Unweighted pair group method with arithmetic mean UPGMA is used to cluster patients with their network similarities as an output. Significance of a cluster is calculated using Fisher-Exact test based on the abundance of cases and controls. The minimum path length was set at 2 and thus including only the pathways of seed proteins connected directly to each other and or via a single intermediary protein imputed genes. A separation test was performed to determine the distance between the PPI networks of the MMR deficient and MMR intact dominated clusters using a Python script adapted from Manchi et al. The Proteinarium dendrogram output reveals significant clusters, and 2 clusters were chosen for further investigation based on degree of domination by either MMR deficient or MMR intact patients and number of patients in the cluster. Those results are shown in Figure 1. The MMR deficient cluster contained 52 MMR deficient patients and 0 MMR intact patients, while the MMR intact cluster contained 51 MMR intact patients and 6 MMR deficient patients. DMP1 was found to be the hub gene present only in MMR intact patients. DMP1 is an extracellular matrix protein critical for the proper mineralization of bone and dentin. In the MMR deficient consensus network was the presence of a subnetwork of 5 histone genes, 4 of which are from the histone H2B family and were unique to MMR deficient patients. The one gene encoding a member of the histone H2A family was imputed. GMB1, which encodes the beta subunit of G proteins, and CDK1, encoding the catalytic subunit of the M phase promoting factor necessary for G1S and G2M phase transitions in the cell cycle, are both highly connected genes present in 39 and 34 of the 52 MMR deficient patients in this cluster. The distance between the MMR deficient and MMR intake consensus networks in the interactome was calculated as 0.1166, indicating no overlap in the interactome and can be regarded as distinct molecular entities.